Medal for academic excellence. With over 29 years of experience in teaching and research, she has guided 80 MPharm and PharmD projects, and 12 of her project works have won the Tamil Nadu Pharmaceutical Sciences Welfare Trust Scholarship Award. She has contributed significantly to pharmaceutical sciences with 53 research publications in national and international journals and has presented over 50 research papers at various conferences. As a resource person, Dr. Manjula Devi has played a key role in numerous professional development programs, including the International Conference on Future of Medicine organized by Tamil Nadu Dr. MGR Medical University, and has led programs on patient counseling, good pharmacy practice and safe medication practices across various institutions. She has also been involved in hands-on training sessions on pharmaceutical care services and drug dosage calculations for faculty, interns and clinical pharmacists. In addition to her academic work, she is a Udemy course instructor offering a certificate course on incompatibility of intravenous medications for healthcare professionals. Her primary research interests include medication safety, pharmacovigilance, AI-enabled medical devices, materiovigilance, IV incompatibility and drug dosage calculations. Dr. Manjula Devi is an active member of several professional organizations, including the Indian Pharmacist Association, All India Pharmacy Teachers Association and Kerala Pharmacy Graduates Association. With the short introductions, I would like to invite Dr. A.S. Manjula Devi as a chairperson. Please, ma'am. So we can wait for a few minutes. Here is a small break. We are waiting for the resource person to come. He has a problem with the PowerPoint, it seems. So please wait for a few minutes. I think all of you are sitting nearby your friends only, isn't it? So we can go for a small fun activity. So let us stretch a bit and wake ourselves up with a fun activity. I am going to ask you all to stand up for a moment, please. Don't worry, it will be very quick and we will have some fun. I want you to turn to the person next to you and give them a high five. சத்தமே கேட்கலாமா? 2 கை தட்டினா நல்லதான். சீக்கிரம் தட்டு. It's a small it's a small gesture, but it brings a smile to everyone. Go ahead and do it. While you are doing that, remember that conference like this are about building connections, and a hi-fi is a simple way to share your positive energy. All right, now that we are all energized, let us continue with our exciting day ahead. Please be seated. Let us resume our session. I invite Dr. A.S. Manjula Devi to introduce our speaker of this session. Please, ma'am.
Thank you, madam, for your nice introduction. Warm good morning. Most respected principal of this institution, Dr. R. Sambat Kumar sir, Vice Principal, Dr. V.S. Saravanan, and organizing secretary of this great event, Dr. C. Kannan, Associate Professor, Department of Pharmacy Practice, distinguished delegates, speaker of today's current session, Mr. R. Rajaguru sir, faculty members, and dear participants, registered participants and students of this institution and volunteers, warm good morning to one and all. At the outset, I thank this institution for giving me this great opportunity to be a part of this wonderful event. And today's session topic is going to be one of the most important topic which we all should update. It is on generic product development for the global pharmaceutical industry. And the speaker of this session is Mr. R. Rajaguru, Manager FRD, Cure Afia Pharmaceutical Limited, Tanzania. And let me take this opportunity to introduce our speaker to you now. Mr. R. Rajaguru, M.Pharm in Pharmaceutics, is an experienced pharmaceutical scientist, currently serving as Manager of Formulation Research and Development at Cure Afia Pharmaceutical Private Limited, Tanzania, East Africa. Sir has earned his Master of Pharmacy degree from Pondicherry University in 2015 to 17 with a strong academic record, following a Bachelor of Pharmacy from Annamalai University in 2011 to 15, where he graduated with 80.6% marks. Sir has professional experience, spans multiple reputable pharmaceutical companies across the globe, including Cure Afia Pharmaceutical in the WHO and domestic markets, Tanzania, Shilpa Medicare Private Limited, USA market, Medrich Labs Private Limited, Japan market, Gravity Pharma Labs Private Limited, US FDA and UK market, and Hetero Labs, US FDA, India. He was honored with prestigious AACTE fellowship during his M.Pharm studies and successfully qualified the Graduate Aptitude Test GPAT 2015 with an All India rank of 125, as well as the NIPER rank of 487. Mr. Rajaguru has actively contributed to pharmaceutical research through over 10 national journal publications and 1 international publication, and multiple conference presentations, including award-winning oral presentations on innovative topics such as fabrication of erythroid stellate fish skin collagen, collagen-coated nanofibrous scaffold as a potential dermal substitute for skin tissue regeneration, and magnetic microspheres as targeted drug delivery systems. He holds a patent also, a UV trans-illuminating device, and he has completed specialized CR-funded programs as the Central Lab Research Institute in specialized CSIR-funded project training at Chennai and Lucknow also, focusing on development of polymeric wound dressing material for tissue injury application. Continuous professional growth Mr. Rajaguru has participated in over 30 seminars and webinars across major Indian states and enhancing his expertise in pharmaceutical formulation, drug delivery systems, and product development. His blend of academic excellence, practical industry experience, and research innovation equips him to make as well as healthcare solutions globally. With this brief introduction, let us invite Mr. R. Rajaguru to kindly.
Good morning. So today we are going to discuss generic product development for global pharmaceutical industry. Okay. So how we are going to make for the generic product? What is the innovator product? Please come here this side. So here is a B pharmacy only there or M pharmacy is there? PharmD also is there. Okay. So today we are going to discuss generic product development for global pharmaceutical industry. So this is a very major topic for especially for B pharmacy as well as M pharmacy, and PharmD for just for fundamental things. Okay. So please, my session will be at least 45 minutes or 40 minutes only. Please listen. So any questions or 100 times you can ask also, I can answer you. Okay. But the question will be valid question only. Okay. So B pharmacy why is a very important means whenever the going for the pharmaceutical industry, so what are the questions they will ask? What is the generic? We are taking generic or innovator. So you should know about the pharmacy student. So M pharmacy, so how you can develop the generic product, we should know. So M pharmacy, how they are developing the products you should know. B pharmacy, what are the process you should know. What is the generic at least you should know. Okay. So please listen carefully. So whatever the dosage form, how we are developing, okay, what is the drug delivery forms, okay. So what are the side effects will be there, how we are developing, how we are approving the product, what are the market will be there, so how to register. So these are the things, fundamental things I am going to discuss. Okay. So if any question is there, if you middle session also you can ask, but you should ask some limited time only. Okay. So please. So this is a main content will be there. So introduction of the pharma. Okay. So pharma company we have the R&D center as well as for the manufacturing plant. How many people are you know about the R&D center? So R&D is a research and development. So we are going to discuss about the what is the research they are doing in a pharma company. Okay. So what are the who are the eligibility? Okay. So who are the branches will be working for the in R&D center. Okay. Manufacturing plant, what are the people are working? Okay. What are the department we have to work? So we should know. So what are the B pharmacy where you have to work? M pharmacy where you have to work? Okay, PharmD, you can interest if you can work, or either you can go for the clinical pharmacist or clinical whatever may be. Okay, so second one is a different dosage form. Third one is a generic product development. Fourth one is a different optimization process. So scale up batch to commercial batch. Whatever we are doing the research of the tablet, small quantity of the tablet to commercialization of the product. For example, I am going to make for research purpose 500 tablet or 1000 tablet or 2000 tablets. So 2000 tablet, what are the challenges will be there? We cannot make for the 500 tablet easy process to 5 lakh. So process will be different. So material will be different. Okay. So equipment will be different. Person will be different. Okay. So we should know how we are going to make for the small tablet, small quantity of the batches to large quantity of the batches. So that is we can call as a small scale batches and commercialization batches. Okay. Fifth one is the calculations. So very important for calculations. Suppose you are missing the some calculation of the quantity of the material or material name, so total product we have to go the discard. Okay. So that's the reason only there mentioned for the any documents, prepared sign, reviewed sign and approved sign. Suppose prepared person something done mistake, so we have to reviewed person should we have to clarify. Suppose reviewed person, suppose if there's something done mistake, so approved person carefully have to do the approve. So these are the fundamental things you should know about before going for leaving the course. Okay. So this is for my content I am going to cover for today. Okay. So generic. So what is the generic? What is the innovator? So what is the cost of the generic? What is the cost of the innovator? So suppose generic will be 25 rupees, innovator will be almost 85 rupees. What is the difference? Anyone know about what is the generic, what is the innovator? I have to tell only one person one. What is the innovator? Any idea? So suppose you are taking the medicine, that is a generic or innovator? So paracetamol will be generic or innovator? How you are saying generic? On what basis you are saying the generic? Paracetamol is a generic. So I will tell that is paracetamol is a innovator. Huh? So what is the paracetamol cost? Anyone know? Paracetamol price. So 1 rupee or 2 rupee or 3 rupee based on the branded they will sell. Okay. So if you are buying the innovator, so that cost will be around 25 rupees one tablets. Okay. So we cannot buy for the innovator here. So wherever the doing for research center or particularly innovator making product, so they will sell for the one tablet of the paracetamol 25 rupees or 30 rupees including the GST. Okay. So this is a different. So we cannot make for the innovator large quantity. I mean we make for the large quantity of the innovator, people cannot buy, cannot afford. Okay. So that's the reason only we are making for the generic product. Same quantity, same qualitative, same material, same manufacturing process. We cannot compromise any for quality of the product compared with the innovator. So what is the innovator will be there? So same you have to make for the generic. We cannot compromise anything. Okay, but you have to make for the low cost of the medicine, but it is a quantity, qualitative, everything should be same, but cost wise, okay. So then only people will can buy, affordably can buy, so we have to cure the disease. Okay, this is the concept we are doing for the India generic product. Okay, so whenever the making the company, so they will mostly focus for the generic. Okay, so we cannot, will buy more as well as manufacturing of more, so getting benefit for company also go fast. So the reason only they most of the company they are looking for the generic product. Okay, so again I tell clearly what innovator will be there, same you have to make for the generic. Okay. So that will be no any differentiation will be there. So you have to make. So whenever we are making the medicine, you should know how will be work. That is exactly they are matching or not. How should we decide? So we have to compare with the innovator. What is the innovator will be their test. For example, assay 98 to 110 or 102. So same you have to make for the generic also. Okay. So we cannot justify it, we cannot escape, nothing we cannot do. Okay. Same what is the innovator is there. So you have to make with the same limit, same parameter, same chemistry test, same microbiology test. Everything you have to make with similar only, but within limit. If 100 means 100 we cannot make, some plus or minus will be there as per the guidelines. Okay. So 98 to 102. So you should limit also, 100 also okay, 99 also okay, or 102 also okay, but it should be within limit. Okay. So this is a generic as well as for the innovator. So what is my goal? So main is a decrease the price. Okay. Second one is so you can off now. No. So whenever the buying the innovator, we cannot sell for the 100%. Why 100% means that cost will be high. So manufacturing process will be high, sophisticated machines. So only they are making for the comparison purpose only. So we are medicine, so the generic medicine comparison purpose they are making for the small quantity only. So they are not make higher quantity because now they cannot afford to sell the people. Okay. So they make for the 50% or 30% only. So once they are making the medicine, so you should sell for the, especially for the innovator, they will not sell for the human. Okay. So some rich people they will buy. That also very rare case only. Okay. So maximum they will spend for the bio-study purpose as well as for the R&D research and purpose. Okay. So this is the difference between the innovator as well as for the generic. Okay. Anyone clearly understand or not? What is the generic? What is the innovator? Okay. So this is the pharma company will be there. What are the pharma company? R&D center and manufacturing plant. Total is different. Okay. Whenever they making the medicine, whenever they making the new medicine or generic product or redevelopment, whatever the may be final formula, so we are sending to the manufacturing plant, they will make the bulk quantity of the medicines. Okay. So what are the department will be there, you should know fundamental things. So major department is major will be there, minor will be there. So major will be formulation and research development, and minor will be analytical research development and development of quality assurance, and another one is packing research and development. Okay. So what is the major department mean? Formulation. So whenever they making the formula, they have to finalize. So the product will be stable on the market. So your final formula will be stable. Your study, bio-study will be have to match, as well as chemical test have to match, physically have to match, everything have to match for the formulation department. So once is a formulation department, they finalize the formula, they will send the QC. QC they will do for the different analysis. Okay. Once this analysis okay, then we are sending for the bio-study. So bio-study normally who are people are will be there, PharmD people will be there, clinical purpose. Okay. They take the sample for feed condition or fast condition, urine sample or blood sample, whatever may be samples are they taking for the PharmD people will be there. Okay. So they will give the result, bio-study report. Once this bio-study is okay, then we have to, product have to register the particular market. I mean particular market means what are the country we are doing, so that country purpose some regulatory body will be there. Okay. So that regulatory body have to register the product. So that regulatory body they approve the product, then we have to start the commercialization. Up to that formula should be in R&D only. Okay. Once it's approved, then they will given to formula to manufacturing plant, they will make the bulk quantity. Okay. So supporting department. So these are the three department will be supporting department. Whenever I am making the formula, someone should be properly have to verify. Okay. So that department is a quality assurance department. Development of quality assurance department. So that people are sitting in R&D center. Second one is analytical research and development. Okay, whatever the chemical test they will do for the analytical research development. Okay, that candidate should be B Pharmacy or M Pharmacy or BSc Chemistry or MSc Chemistry. Okay, so M Pharmacy should be pharmaceutical chemistry or pharmaceutical analysis. Okay, so that people they will work. But in formulation department only we can work only one people only. That people is pharmaceutics department only. But is a chemistry, QC quality control department, either is a B Pharmacy or M Pharmacy or BSc Chemistry or MSc Chemistry should work. But formulation we cannot work for all department, only the department only work for the pharmaceutics only. Okay, they will not take for all peoples. Okay, so that is the reason only their major department they are kept for the formulation and research development. Okay, second one is the manufacturing plant. So manufacturing plant, what are the department will be there? Production department will be there, regulatory department will be there, quality assurance department will be there. Different between the development of quality assurance and normal quality assurance. Okay. Third one is a quality control department. Sorry, fourth one is a quality control department. Fifth one is a microbiology department. Sixth one is a warehouse. Seventh one is a engineering, and eighth one is a marketing department. Okay. So these are the manufacturing plant departments. Okay. So every department should be very, very important role. Every department should work only, then only the medicine should be come out for the manufacturing plant to patient or wholesaler or medical shop. Okay. So this is the fundamental things for the, what is the pharma company, how they manufacturing of the medicines, what are the department their working role, so you should know. So especially what is the B Pharmacy, where should work. Okay. Either you can work production area, either you can work QC, either you can work microbiology. Okay. So up to B Pharmacy you are completed means, if you are getting job means, you can work. Some people you can work also regulatory. Anyone know about regulatory? What is the regulatory body? So what is the regulatory body means? So whenever they are making the medicines, so someone should be registered. Okay. If for example you are writing the exams, so examiner will be there. Same like that regulatory. So I am making the medicines, so I am making the documents, so I am sending to regulatory body. So regulatory body, particular regulatory body, they are uploading the regulatory departments. Okay, government body. So that department is called as a regulatory department. Okay. So they verify the all documents, they verify the parameters, every department documents, not only F&D, every department documents, they verify the data, they verify the everything, theory, data, practically everything they verify, they upload to government body. Once it's a government body, they review the documents, they approve the medicines. Okay. So normally B Pharmacy also we can get regulatory, but it's a very, very less chances will be there. So some technically knowledge should be there. Okay. So what is the product, how we have to register, what is the regulatory body, how we have to register, fully system work only. No lab work, nothing will be there, only fully system work like a software work. Okay, regulatory body. So we have to upload the documents, so we have to review the documents. Okay, how there any mistake will be there, everything you have to find out. So final touch up will be regulatory department. Okay. So this is a very major department. You can work some of the getting job also, B Pharmacy student also, you can get job for the regulatory also. Okay. So this is a different dosage form will be there, everyone know? Okay. So dispensing pharmacy you can study what is the tablet, what is the capsule, granules, powder, suppositories. So everything will be there, you should know. Okay. What is the solid dosage form, what is the liquid dosage form, what is the semi-solid dosage form. Okay. So I can ask only one question. If you are clearly you are studied for dispensing pharmacy, mean you can answer. Okay. So anyone tell me what is the capsule? Any product name you tell me? Capsule product. No, it should be answer one person only. If you right, wrong, no problem. But so answer will be one person only. Okay. Anyone tell me capsule names? Any capsule, no problem. Antibiotic, whatever may be, cancer, antibiotic, whatever may be. Tell me. Okay. Amapsin. Okay. Omeprazole. So omeprazole is a proton pump inhibitor. Correct? Pantoprazole, omeprazole, esomeprazole, lansoprazole, these are the proton pump inhibitor. So anyone proton pump inhibitor, how they are dissolving the stomach. If you really they are dissolving the stomach, we should know one thing. And one more thing, omeprazole capsule is there. Inside what is there? Either is a pellets or powder or tablets. Pellets. Okay. So amoxicillin. So you are taking the capsule, correct? Hard gelatin capsule, not soft gelatin capsule, correct? Hard gelatin capsule, omeprazole. So they are filled in the pellets. Amoxicillin they fill in the powder. Okay. Why? What is the difference? Okay. So anyone see the capsule filled in the tablets? Okay. So mycophenolate meftil, okay, mycophenolate meftil, that is a immunosuppressant, okay, that category is a immuno, okay. So that capsule is contain mini tablets, okay, mini tablet of the capsule, okay. So these are the things we are doing for the innovations, okay, just an example I am telling. So normally where people are buying the amoxicillin, pantoprazole, omeprazole, esomeprazole, everything we are buying, but so some new technology, some new innovation we are doing mean these things only. Okay. So we are filling in the capsule in tablets. Not like a paracetamol tablets, that size of tablet, mini tablet, 5.5 mm. I mean this much size, small size. We are filling in the capsule. Okay. And one more thing, another things we are filling, granules. I mean granules means how will be there physically? So powder should be without moisture, correct? So powder should be without moisture. If you granules mean with moisture, that is called as a granules. Okay. So we are adding the powder, some quantity of the water mean that is called as a granules. Okay. If you are going to dry means dried granules, without drying means that is a granules. Okay. So dried granules we are filling in the capsule. So ultimately how many capsule we are doing? 4 capsule we are doing. One is a powder, another one is a pellets, another one is a mini tablets, another one is a granules. Okay. So these are the things, this outside is a new innovation is going on. So these are things how should we update. Okay. We have to read the articles only. Okay. Whenever the pharma, any update or any news or any articles means you should learn like that way only. Don't have any other options. Okay. So nowadays is coming new product also, paracetamol oral film. Paracetamol oral dispersible film. With a second you can reach the systemic circulations. With a second. If you are kicking the tongue, not for the downside, upside, with a second they will dissolve. Okay. Normally paracetamol should be very bitter or not bitter. So we are challenging with bitter only. Okay, paracetamol highly bitter. API is a highly bitter, but we are keeping your tongue. How much bitter will be there? Just imagine. Normally we are taking the tablet with water. Just they are touching the mouth. It is too much bitter. Correct. So raw film. So we are keeping tongue. Mean how much bitter will be there? Just imagine. So without bitter we have to do. Okay. So we have to do for the different flavor optimization. Okay. So these are the things are going on for research, okay, outside. So you should know about everything should be update, okay. So how should update? Pharma journals or research paper, review paper or patent should be, you have to read, then you get it knowledge for the fully, okay. So these are the major we are selling for the market. So tablet should be 78%, capsule will be 48%. This what not only India, this is globals, okay. So injection will be 24%, syrup is a 2%, suppository is a 4-6%, posterior is 12%, and cream or cold is a 9%, suspension is a 0.3%. So these are the percentage we are selling for the medicines. So maximum we are focusing for the tablets. Okay. So nowadays we are making for the oral dispersible film, nanotechnology like. Okay. So nanotechnology we cannot sell, but we have to do for the research work. But we are doing for the research as well as we have to sell. Okay. So nowadays technology is going on like that. So another dosage form, it is come under the oral dispersible film. Okay. So suppose we are going to do for the new product development. What are the requirement is required? Okay. One is a active pharmaceutical ingredient. What is the active pharmaceutical ingredient? Main product means? Okay. So paracetamol not required. Paracetamol no. Any other product we can tell. Okay. For example, cetirizine. Okay. Cetirizine is a active pharmaceutical ingredient. Cetirizine hydrochloride or cetirizine dihydrochloride. Okay. So that will be active pharmaceutical ingredient. Third one is excipients. We cannot take for the 100% of the active pharmaceutical ingredients. Okay. So we have to make some excipients. Either is a diluent, either is a filler, either is a disintegration, either is a super disintegration, either is a coating material, either is a talc. So many things will be their category. We cannot take for the 100% of the API. Okay. Suppose we are going to make for the some food. Okay. Gravy. So that main content will be gravy, but it's additional quantity we are adding. Same like that way you have to add for the medicines. So that are the whatever the material we are adding, we have to justify how much quantity you have to add, how much percentage you have to add, everything you should know. Okay, simply we cannot put 1 spoon or 2 spoon, not like that. Every material, every experience, every raw material should be, some percentage will be there, some limit will be there. If you are crossing the limit, ultimately your product will be fail or some issue will come before filing the product. Okay, so experience is a main important role. Third one is a manufacturing process. So you have the API, you have the experience, so we have to manufacturing, how to manufacturing the process. Okay. Fourth one is a drug delivery form. So whatever the API, whatever the raw material, whatever the manufacturing process, so once it's a manufacturing process, the tablet should be, you have to take. Which drug delivery system, which form you have to take. Okay. Which delivery you have to take, which dosage form you have to take, you have to decide. So these are the 4 things required for the how they are developing the new product. So majorly I work for the US market, Europe market and Canada market and Japan market. So mostly I worked for this foreign country market only. Till now I not work for Indian markets. Okay. So my company fully for the highly big company. So I work only focus for the US market or Europe market, Japan market, Canada market only. Okay. So these are the regulatory body. So US means United States of the Food Drug Administration. So that is a regulatory body. You have to file the regulatory body of the medicines. Whatever the making the product, you have to file that regulatory body. UK means that regulatory body. Canada means Health Canada. So you should know about what is the regulatory body, what is the market we are working. That market you have to register of the product. Okay. So we have to follow means some guidelines is required. Fundamental guidelines you have to refer for the ICH guidelines. Okay, that is a globally common guidelines for the ICH guidelines. So requirement for generic. So I told you first innovator. So innovator similarly you have to make for the generic. Okay. So just fundamental things I have to share you. Same active ingredient, same route of administration, same strength, same dosage form, same condition to use. For example, someone you can ask me question. If I am changing something, everything I written first letter same, correct? Same route of administration, same active ingredient, same condition. Okay, every file content I written for the same, correct? So if you suppose you are changing something, so I am going to change for the strength. For example, paracetamol 500 mg I have, I am going to change for the 750 mg. Okay. So we can change like that. We'll change. Paracetamol have 500 mg. So you want to make for the 750 mg. So already we have the 650 mg. So you want to change for the 750 mg or 450 mg. Okay. We can change. Huh? Will change or not? So suppose if you changing means what is the impact will be there, what is the process you have to do. If you not changing means what is the process you have to do. Okay. Suppose you are going to change. If you are going to change mean that is a innovator. Okay, if you are changing means you should be have to do for the all studies. Okay, so including the animal study, including the human study, everything you have to data you have to submit to particular regulatory body. If you are properly you are done every studies, so you can register. That will be patent you can file. Up to how many years, that will be your product only. Okay, but that document should be properly, every evidence should be will be there. Okay, bio study as well as animal study, every study for as well as for QC, micro, every study you have to conduct. Up to literature too, up to registered, so many study will be there. That is, we cannot change for within one day. Okay, it will take for the 1 year or 2 year or 3 years also. Okay, stability data, bio study, drug expense compatibility study. Okay, large scale, small scale. So so many study will be there. Within, we cannot do for within 1 month. It will take for 3 years or 2 years or 4 years. Okay, so we can change it. So if you are changing me, we have to properly justify the particular regulatory body. We have to properly have to do for the some studies. Okay, so we can change it. So same, all should be have to same you have to follow. Okay, as per the innovator, what is the innovator will be there, same you have to do with the limit, we have to file the product. Okay. So what is the ANDA versus NDA? I told you, you can change it. Okay, 450 mg. So 450 mg you can change it means that is called as NDA molecules. Okay, new drug applications. If ANDA means abbreviated new drug applications. Someone they filed the product, someone same strength, someone same condition, someone same category, someone same active ingredients. So you are doing same. Okay, so that will be abbreviated new drug applications. If you are someone did, so you are deviating, you are doing something innovator, so that called as NDA molecules. Okay. So what is the difference between NDA as well as ANDA? Okay, please write it, this NDA versus ANDA. Whenever they are going the examination, GPAT exam or NIPER exam or government exam, they will ask for the what is the NDA molecules, what is the ANDA molecules. Okay, you should know about the abbreviation of the ANDA plus NDA. So what is the requirement for the ANDA, what is the requirement for the NDA molecules? ANDA is the dispensing, chemistry, manufacturing, controlling, labeling, okay, testing, animal study, the chemical study and bioavailability study. I told you new product we are going to register means you should record for the animal study as well as for the clinical study, I mean human study. Okay, so every condition, human study means every condition you have to take. Okay, before meals, after meals, urine sample, blood sample, okay, everything you have to conduct. Voluntary, non-voluntary we cannot do, voluntary we have to do. Okay, so whenever we are, suppose some category we are doing mean you have to do for some survey for the some category. If diabetic we are doing means so many research institute can go there, you have to collect the data, we have to do it. You have to properly justify that category of the product. So we are going to change the strength. So this strength we are going to change, this much drug we are receiving, like that you have to AUC, Cmax, Tmax we can study, correct? Biopharmaceutical pharmacokinetic, Brahmankar book. Okay, this is a very simple book, you can read. So you will get some idea how much drug they are dissolved in the body, how much drug they eliminated, how much drug is metabolized. So you should know. I mean 3rd year or 2nd year it will start, biopharmaceutical pharmacokinetics. Okay, you can study. So suppose we are going to do for the NDA molecules. So NDA molecules, this much required. If you are going to do for the ANDA molecules, I told you ANDA molecule you can, someone did. So same you have to do. Okay, someone did means their formula you can bring and you cannot do like that. Okay, you have to do. So their machine is different, their material is different, your material is different, your scientists will be different, their scientists will be different. Okay, so you should make similar to same condition, similar to same strength, similar to same everything, active ingredient. Okay, so that time so not required for the animal study. Okay, human study is required, animal study is not required. So we can refer for the NDA molecules. So NDA molecule data you can refer to ANDA molecules. Okay, so biostudy compulsory required for the some of the category, biostudy is required, but biostudy some conditions should be there. Okay, for example, someone we have to do for the bio study, with milk we can do, with juice we can do, okay, without food, with food, so many data will be there, okay, so that will be very big chapter, okay, so that things should be refer, everything have to refer, everything test you have to do, okay, so with milk how the drug will be dissolved, with water how they dissolve, with chemicals how they dissolve, hydrochloric acid how they dissolve, okay, so with the sodium lauride sulfate how they dissolve, so every test you have to do bio study similarly, okay, so not required for the animal study, only required for the human study. So I can ask only one question, proton pump inhibitor, again I come to back, okay, proton pump inhibitor, it will dissolve in stomach or not? How many people are taken for a proton pump inhibitor? Okay, suppose we are taking the proton pump inhibitor, it will dissolve in the stomach or not? Pantoprazole or omeprazole or esomeprazole 20 mg or esomeprazole 40 mg, lansoprazole, whatever the proton pump inhibitor, that proton pump inhibitor it will dissolve in the stomach or not? Tablet should be dissolved in stomach, correct? Correct or wrong? Yes. So proton pump inhibitor is dissolving in stomach or not? So please you can go there today, end of the session I will answer, but today please you can go there, proton pump inhibitor, how the dissolve in stomach, how the reacting of the product, how much time it will take. So because we are going very severe pain of the okay stomach, immediately how you got to act, correct? Someone pain, okay, you can go in the hospital, they are giving the proton inhibitor, how much time it is work, how much time will take, okay, where is dissolving the tablets, so you should know. So simple things, question only they will ask for the pharma company whenever they are taking interviews, very simple questions. After interview you came back to outside, you are seeing the mobile, you can laugh, okay, நீங்க சிரிப்பீங்க, ஓ இந்த question தான் நம்மள கேட்டாங்களா அப்படினு, okay, so you should know before. These are the simple questions only, okay, so please you can go there, you can read proton pump inhibitor, how they dissolving the stomach or not, how they are reacting, where is the reactant, so everything you should know proton pump inhibitor today, okay. So free formulation study, okay, so whenever I am going to making the medicine, before I should know about the some fundamental things free formulation study, okay. So B Pharma student as well as M Pharma student, what is the free formulation study, why required, how you have to do, okay, what are the tests you have to do, everything required for the free formulation study. Deeply clearly they will give the free formulation study. First year they will study for the physical pharmacy, correct? So Manavalan one book will be there, my professor only. So that book will be around red color will be there, around 250 pages will be there, okay. So that book clearly they will mention what is the physical pharmacy, okay, that powder should be what are the physical characterization we are doing, okay. So I am giving you one powder, okay, API will be white color, raw material will be white color, so you should know, okay, how the crystal form will be there. So this crystal form means what is the material name, okay, what is the grade name, okay. Suppose amorphous means how should know, every product, every material should be white color excepted coating material, correct? So coating material different color, but APA and raw material is a same white color only, but same white color, but its a form will be different. I mean crystal form, amorphous forms, okay. So crystal form means like a sharp color, some crystal will be there, okay. So amorphous form and powder form. Amorphous form will like a powder, face powder is like a smoothness, okay. Powder form means normal powder only, okay. So we should know with that powder only we should know what is the material name, what is the grade name. So grade wise they will change, okay. Grade wise the physical characterization, that microbial and particle size distribution, everything will be different. So that study is required for the physically as well as chemically, okay. So physically what are the tests will be there, crystal form or amorphous form, okay, hydrophobicity, particle size distribution and powder flow properties, okay. What is the powder flow properties means, suppose you have the powder, correct, raw material, So raw material, 2-3 raw material you have. API we have, raw material 2-3 raw material we have. So we are both mixing, so we are pouring to the compression machines. Okay, compression machine means tablet punching machines. Okay, so we are adding the 100 kg or 200 kg in the machines. So powder should be done in flow. Then only machine should be running very fastly. Okay, that flow purpose we have to do for some studies. Okay, so each and study, every study we have to study as well as we have to results also get it done. Okay, then only we have to send the formula to manufacturing. Suppose I am taking the 200 tablet for the R&D center, the tablet flow will be good. If you are going to the manufacturing plant, so the tablet will be, powder will be stucking the machines. So that stucking machine means I should know about the why they are stucking the machines. We are going to add the lubricants. We have to add the glidants. So that are things we have to finalize. So simply we cannot take powder into the compression machine, we cannot start. Okay. So every data physically we have to identify. Suppose tablet we are doing the punching the machines, some capping issue will come, some sticking issue will come. Okay. So some cracking issue will come. So everything have to resolve. Okay. So this tablet is coming to the market with very small size as well as with finishing means so many persons backside will be there. Okay. So we have to finalize the, like that way, we have to finalize the formula. That formula should be very strong in the R&D, then only we have to send the productions. So production once started means same formula only we have to, how many years they are approved the product, up to that formula can continue for the market. Okay. So physically we are doing, chemically what is the, we are going to do. Solubility study, okay, pH effect, thermal effect, solubilization, partition coefficient study, dissolution, stability, solubility, pH rate profile, solid rate, bulk stability, compatibility. Okay. So these are the things we have to do. These are things for the main things for second year or third year you are studying. Please can study very deeply. Okay. What is the compatibility? What is the stability? What is the partition coefficients? Okay. What is the dissolution? How they dissolve the drugs? What we are, suppose we are taking the water. Okay. So how we are going to dissolve? Okay. How much time will they dissolve? Okay. So these things you can record for the free formulation study. Okay. So once making the powder to the tablets, so how they dissolve physically as well as chemically. So physically I will check these are the parameters. Chemically they will check for the result. Okay. Some chemical they will add the hydrochloric acid. Suppose I am putting the hydrochloric acid. Okay. So how much time it will take the dissolve. Okay. If you are adding the proton pump inhibitor in hydrochloric acid, how much time will take. So you should know physically as well as chemically. Okay. So this study before you are going to the making the formula, finalize the formula, before we have to do for the free formulation study. Okay. Okay. Product development chart. Okay. This will be Google openly is available. Product development chart. Okay. Suppose we are going to do for the new product. These are the step is compulsory is required. It is ANDA molecules or NDA molecules, whatever the molecules. So compulsory you have to follow only. We have to do only. So who will do? R&D person will do. F&D persons they will do. That sample should be sending for the QC or analytical research development or microbiology. We are sending the sample to the supporting department. They will give the result. So we are compiling. So that the compiling documents as well as physical document we are going to give the regulatory body. That regulatory body should be registered the particular regulatory departments. Okay. So one is the literature searching. Another one is the active source, active evaluation, active purchasing, active testing, innovator product purchasing, innovator product testing, bulk active testing, experience evaluations, container closure systems, manufacturing process, bulk active purchasing, analytical evaluation, process optimization, scale-up batch, process qualification, pivotal bio batch, A&A pre-submission audit, A&A submission, product development report, and process validations and process revalidation. After that, major changing the SUPA guidelines we have to submit for the commercializations. Okay. So one by one I will tell you. Suppose you are going to make the new formula or going to new product. Suppose you are going to work R&D center. Okay. So they will give the new project or 3 or 2 product they will give. They will tell the names. Okay. So these are the product you are going to do. Okay. They will giving the pantoprazole or they giving the omeprazole. They will give the strength as well as pantoprazole or omeprazole. Okay. So what you have to do? First you have to do the literature. Literature means whatever that product should be, everything you have to take the data. How we are going to take the data? Literature means review article, research article, patent. Okay. So these are the documents should be collect, you have to read. Okay. So what are the tests they are done? What are the chemical tests they are done? What is the difficulty will be there? Physically any difficulty will be there? Chemically any difficulty will be there? You should know. How should know? You have to read the journals. You have to read the articles, review article or research article or patent. Okay. So some of the patent they will open, some of the patent will be not open. Okay. So open patent you have to read. Same like that research, some research they will close, some research will be they not close, open. So you have to download, you have to read, you have to collect the data, you have to compile the one documents, we have to submit to the your manager. So your manager will be review that data. So then we have to start the procurement, we have to start the manufacturing process. Okay. Active source means API. We have to purchase the API. Okay. So what product they are allotted? They are allotted for the 2 product. Okay. So the 2 product, what active pharmaceutical ingredient you should know, correct? Omeprazole is a active pharmaceutical ingredient. So that active pharmaceutical ingredient we have to purchase. Okay. So that purchase, after purchasing you are receiving the material should be hard to evaluate. Okay. Physically as well as chemically. Physically how we are evaluate? Free formulation study. Chemically how we are evaluated? So that sample sent to the QC or analytical research development. Okay. They will check the chemically. Okay. Physically you should do for formulation department. Okay. So next one is a active purchasing, active evaluation we are doing. Another one is a testing are doing, and innovator we are purchasing. So okay. So innovator whenever they making the formula, so we are comparison. Correct? I told you comparison. So comparison purpose, please. Another 20 minutes I will close. Okay. Please. So same like that only. I am also so many conference I went. Okay. So same it will sometime it is bore only. Okay. So please. So I am traveling almost 17 hours. Okay. Not only 1 hour, 2 hour, 17 hour by flight. Okay. Please. Don't waste my time. So I need to go for the another session also. So please. Only 20 minutes I will close. Okay. So innovator we have to purchase. That innovator should be have to test. Okay. The test physically as well as chemically. So both you have to do for the comparison. Okay. So every test, whatever the stage you have to do for the testing as well as evaluated you have to do. What is the container closure system is called as a packing material. Container closure system is called as a packing material. Okay. So that name will be how will be the, what is the container closure system means they will ask interview questions. Okay. Container closure system is called as a packing material. Packing material means so many packing material will be there. Okay. Aluminium foil, alu foil, BVC blister, PVDC blister, container, HDP bottle, high density polyethylene, loose and dyed, okay, brown color HDP bottles. So this many things will be there, please. Okay, why I am telling this many things means whenever the going interview definitely they will ask, whenever the writing exam they will ask. For example, PVC, PVDC, why we are packing the material. For example, paracetamol, okay. Paracetamol, what is the packing material? Anyone know? Before B pharmacy also, we are buying the paracetamol, correct? So we are taking paracetamol up to childhood onwards, correct? So what is the packing material? So this is the interview questions. Okay. Openly I am telling this is the interview question. They will ask what is the paracetamol packing material. So how we are feeling? You are not answering me how we are feeling. we are feeling very guilty only. Why? Because we are studied 4 years pharmacy, and before pharmacy you are taken for the paracetamol, correct? So what is the packing material? They will ask simply. So that's the reason only I am saying so many background will be there, everything. Okay, whatever we are doing, so many things will be there. Okay, so whenever the confidence, whenever the reading the articles, so if you are reading very clearly, so you can answer very clearly to interview persons. Okay, so paracetamol is a upside will be the PVC. Okay, clear PVC. It is called as a clear PVC. Downside is a aluminium foil. Okay, the backside will be the written is a paracetamol tablet 500 mg. That will be aluminium foil. Okay, upside the covering is a clear PVC. What is the PVC? Okay, good. So upside will be clear PVC. Okay, downside will be aluminium foil. Okay, for example, both side will be there aluminium foil. Okay, pantoprazole tablet 40 mg, DR tablet. Okay, delayed release tablet. You can see there both side will be aluminium foil. Aluminium foil will be there, not PVC. Upside also aluminium, downside also aluminium. That is aluminium, hard aluminium. Okay, so you should know packing material. Container closure system is called as a packing material. Okay, so packing material is a very important role in the product. Okay, whenever they making the formula, whenever the manufacturing of the product, you should be stable in the market, should be stable in the medical shop, or should be stable in the warehouse. Based on your packing material, based on your container closure system, we are properly you have to maintain, properly you have to do for the test. Then only your product will be without contamination, without any issue, it will stable in the market. Okay, so how we are deciding the 3 years, 2 years? Okay, some of the product we are deciding the 3 years, some of the product we are deciding the 2 years. So some reason is there, correct? Exactly why they are keeping the 2 years, exactly why they are keeping in the 3 years. How? Means based on your stability data. Okay, please. Based on the stability data, so you should know about the product stable in the market. Okay, so stable, how is stable means, so we have to load the stability. Okay, stability, we have to stability chamber we have to load. So 3 months or 6 months or 12 months or 36 months or 24 months we have to load your product in the stability chamber. Okay, so stability chamber should be, suppose you are making the final formula, we have to load the stability. Okay, so stability, different condition zone will be there. For example, we are in which zone? We are in cool zone or hot zone, or which zone we are? Hot. So what is the different zone will be there? How many zone will be there? The US is a same India zone. What is the zone of USA? Say what is the zone of US for India? So please read. Okay, what is the zone of India? Okay, maybe something in this session will be had some clarity from your side. Okay, so many things we have to read. Okay, whenever the reading the books, whenever the reading the manual, should be read properly, clearly. Okay, then only you can answer properly. Okay, maximum 99.9% they will ask interview people manual only. Okay, so manual you have to read very properly, very clearly with understand. Okay, so you should have to learn. Okay, so we are in climate zone in 4th, hot zone. Okay, China is a zone 1. So if you hot zone, how we are going to pack the material, how they pack the medicines. If your zone will be first zone, China. Okay, so how we are going to pack? Okay, so we cannot pack for the aluminium foil for the zone 1. Okay, so I have to pack for the HDP bottles, high density polyethylene bottle. That bottle also different type will be there. Okay, so one is a hot, another one is a loose. Whenever the buying the medicine for vitamins, So vitamin they will load for the HTP bottles. Thyroxine, okay, whenever they are getting the thyroid, so they will pack in the HTP bottles. That HTP bottle also loose as well as tight. How we are finding? Okay, if a HTP bottle you can center, you can push means it will going loose means that is a loose HTP bottles. If you cannot tight, the tight means that is a hard HTP bottles. Okay, so suppose your product will be light sensitive of the product, so you have to pack for the some brown color aluminum foil with PVC foil. Some of the product will be in brown color foils. Okay, so why that reason means that will be light sensitive of the product. Okay, if you are taking the directly with the light, so that will be drug will be API will be, so it will affect. So it will getting the release also very less. Okay, so many things will be there. So suppose someone will ask how we are manufacturing without light, correct? So without light only we are doing, that will light will be amber light we can call. Okay, that brown color light will be there. So we are fixing the brown color light, we are manufacturing that inside, not for like that light. Okay, white color light, brown color light. Suppose your product will be sensitive or light sensitive product, so do you have to manufacturing for the ample color light. Whatever the dispensing to optic testing also you should be do for the ample color light. Suppose your product will be light sensitive of the product. Okay, so these are the container closure system will be there. So you should know. So we have to make for the bulk product. So that product we have to register either is the A&A molecules or NDA molecules. Whatever the data should finalized, that is called as a product development report. Up to literature to up to whatever the submission data, QC data, micro data, F&D data, all data you have to compile, you have to make some data like a thesis, right? Finally your thesis or M.Pharm thesis we are making one book, correct? So we cannot make like a book, okay? So we are making like a document. So the documents are to review, that document should be uploaded in the particular regulatory body. So that name is a product development report. Every GIST will be there. One product, every product. So all data will be there. That also, this many pages have to compulsory required, like that is there, guidelines. Okay. For example, US market, almost more than 100 pages should be there. Okay, your research work document should be more than 100 pages. Then only they will have to accept the product. Simple things I am telling. Okay. So many things will be there. So this is one of the things. So product development, we have to upload the documents, they will register, they will give the approval for the product. So then we have to start for the commercialization. Okay. So validations. So validation you can start, validation batch you can call also, consider as a commercialization batch also. Okay. So first I am taking for the small scale of the batch. Okay. So I am making for the 500 tablet or 1000 tablet or 2000 tablets. That tablet cannot sell. Okay, only for testing purpose, R&D testing purpose. So that testing tablet we are loading for the stability. Okay, so next I am going to take for the scale-up batch. Okay, 500 tablet I am taking physical parameter, chemical parameter, everything matching. So I am going to take for the scale-up batch. So batch size should be 1 lakhs to 3 lakhs. How will be their physical behavior as well as chemical behavior? Okay. So sometime it will come failure also, we have to overcome. Okay. So that material also we have to load for the stability. Next one is the exhibit batch. So exhibit batch 3 lakhs to 5 lakhs I am going to make. So same physical parameter as well as chemical parameter we have to match. That also I am going to load for the stability. Next is the validation batch. So that validation batch, so whatever the scale-up batch, exhibit batch data, everything is okay. Ultimately validation batch also it is okay only. Because of whatever the formula, whatever the manufacturing process, whatever the BMR, whatever everything I am going to do for the validation batch. Or validation batch you can call as a commercialization batch. So that validation batch you can pass, then only we have to go for the commercialization batch, we can release the market. So this is within not 1 month or 3 months, not 2 months, it will take for the NDA molecules, A&A molecules mean it will take for the 1 year. 1 year you have to do the studies, then only they will release the product. So paracetamol we are going simply we are buying and we are taking, correct? So that work will be more than 1 year work, up to packing, up to data, registrations, coming to the market, coming to the wholesaler, after that distributor, after that medical shop. So this many process will be there within not 1 month, it will take for the 1 year. Okay, if you are going to make for the same active pharmaceutical ingredient, whatever may be, it will take for the 1 year. Why required 1 year? This many advantages will be there, this many sophisticated machine will be there, this many scientists will be there, this many educated people is there. Why taking 1 year? I means 1 year should be every test we have to conduct. That's the reason required for the this many years is taking. If you are going to make new product, so it will take 3 years also. Normally cancer medicine we are going to make, so it will take for the 3 years. So many studies we have to make, separate lab required, this many people only allowed. So many restricted will be there oncology product. Okay, so these are the things will be there. So free formulation study, this is the data for the registration purpose we are going to do for the some data, correct? I told you F&D purpose, MFR, master formula card. So whatever the making the formula, so we have to capture the some documents, what is the formula, what is the API, what is the raw material, what is the coating material, how much mg we are going to take, how much batch size we are going to take, how much percentage will be there. So everything have to capture the one documents. That document is called as a master formula card. Okay, so you have to finalized. F&D% should be finalized. Okay, so same like that packing card. We are going to do for the packing research also. So simply we cannot take for aluminum foil, PVC foil, HD bottle we cannot take. So whenever the making formula, whenever the making the medicine, so I load the packing material, I load to the stability. That stability should be pass. Aluminum foil I load, the stability physically as well as chemically should be pass. Then I have to register the product. If I am going to make the HDP bottle, some microbial growth, some chemical contamination is happen, so I will reject the HDP bottle, I am going for the PVC pack. PVC also fail, I am going to make for the aluminum foil. So these are the study you have to do for the packing, simple packing purpose also. Okay. So that is a primary packing. Secondary packing is called as a cotton box. Okay. So with a cotton box, that is not required for any research work. In a primary pack, compulsory required for the some research. Secondary pack is not required for some research and all. So whatever the data you have to capture, whatever the label claim, whatever the storage condition, manufacturing date, expiry date, okay, which market we are going to do, what is the product name, what is the generic name, what is your brand name, what is the MG, what is the pack size, should be made for the secondary pack. I mean cotton box. Okay, that is not required any stability data, not required for any research. But it's a primary pack should be required for the some research, some backup data. Okay. Second one is a drug experience compatibility study. So you are making the raw material as well as APA should be matching or not. How you can decide? Okay. So your APA you are taking omeprazole, some raw material you are adding. Okay. So properly how they mixing? Okay. You are properly mixing, but chemically how you should know? Some test required. Correct. So identification test. Okay. So we have to take from the chemistry. Okay. They will give the result. So this raw material with that APA. So properly mixing. So we can go for the formula, go for the manufacturing process. So these are the data should be required for the drug experience compatibility study. How your drug behavior with your raw material. Okay. So next is the stability data. Whatever we are doing, same we are going, I told you, whatever the material we are taking, whatever the manufacturing we are taking, going load for the stability. Third one is a B study. Okay. BCS classification study, biopharmaceutical pharmacokinetics. Okay. So we have to do for the some studies. Okay. CARS index. Okay. Sorry. Cmax, Tmax. Okay. AUC. These are study you have to do for the B studies. Okay. So this data is compulsory required. Feasibility test report. What is the feasibility report means? How we are manufacturing the 500 tablet to 3 lakhs tablets. So machine different will be safe, different, correct? So there we are making the 2 liter, we are going, the plant means 5,000 liter. That machine capacity is a big. Here machine capacity is small. How we are going to change? How we are going to challenge with that machines? So that before you have to do for the feasibility report. Okay. So these are the required. So dosage form submissions. So whatever the data, these are all data we have to submit with the dossiers. Whatever the F&D data, whatever the QC data, whatever the micro data, whatever the stability data, whatever the drug experience compatibility data, every data should be compiled with the product development report. You have to incorporate with the product development report. We have to submit to the particular regulatory body. So these are the manufacturing process. Okay. So green color box will be the active pharmaceutical ingredient. How they are making the active pharmaceutical ingredient. Okay. So remaining part will be the how they manufacturing of the product. Okay. So we are going to make them. What are the machine will be there? I am telling you. Okay. One is the dispensing. Okay. We are making for the scoop or some spoons. Okay. Spatula. Okay. Next we are going to make for the some process. Material we have to do for the shifting. That machine will be called as a vibrate shifter. Okay. We are going to load for the some machines. Okay. Two things will be there. One is a wet granulation method, another one is a DC method. Anyone read wet granulation, direct compression method? Wet granulation method means some raw material will be there. We are going to add for the some binders. Correct? Either is organic solvent or water solvent, aqueous solvent. Okay. So that solvent we are adding, we are making for the granules. That is called as a wet granulation method. Without adding water, we are going to make tablet mean, that is a DC method. Okay. So DC method mean what are the equipments we have to handle. Okay. Blender, after mixing the blender, we are going to make for the compressions. Okay. Then if we have the coating, we are going to do for the coating and packing. Okay. This is for the DC method. If we are going wet granulation method. Okay. So wet granulation method, shifting, RMG, rapid mixer granulated. So with aqueous compound with powder we are going to mix, not manually, we have to mix with some machines. That machine is called as a rapid mixer granulator. After mixing we are going to make for the drying. Correct? Drying is a fluid bed dryer. Okay, we are going to make for the drying. After drying we are going to make for the some granules will be there, some powder will be there. We have to make some uniform. Correct? So some uniform will be there, then only we have to make for the compression with the flow properly. Correct? So we have to do for some milling. Whatever the granules, big granules will be there, so we have to break the particles, so you can make the uniform the powders. Okay. So that powder is a multi-mill. After multi-mill we are going to make the blending. Okay. After blending, so we have to start the compression and coating and packing. Okay. This is a wet granulation method. Another one is a DC method. So green color box will be active pharmaceutical ingredient. This remaining white color box will be there. So these are the machine manufacturing process we are going to do for the wet granulation as well as DC method. So I told you calculation, correct? So calculation upside machine, the R&D machine like that and F&D machine like that. Okay. R&D machine very small scale. Another only 3 slide, please. R&D machine will be 2 liter, 5 liter and 10 liter. Okay. If you are going to make for the plant 200 liter, 400 liter, 600 liter, 1000 liter. You can go inside and sit and we can do also like that machine big capacity. Okay. So R&D will be like a bowl, tiffin box bowl. Okay. Like that also we have. Okay. So how will be the, I told you feasibility report. Small 2 liter to 400 liters. So working principle is different. Correct. Machine will be same, company will be same, but the principle will be different, working capacity will be different, loading capacity will be different. Okay, unit operation we can study, unit operation 1 and 2, whatever the machine I am talking, so it will cover. Okay, whenever the reading the unit operation, okay, you can read properly, what are the equipments will be there. Okay, so everything they mention clearly, they will mention multi-mill, what is the working principle, what is the advantages, what is the disadvantages. So which book you have to refer? Unit operation 1 and unit operation 2. I forgot one small book will be there, very small book only. I think it's a 150 or 200 page will be there. Okay, unit operation 1 and 2. Book color will be same, both color will be same, even though 200 pages only. So you can read very carefully, that working capacity will be there, everything they clearly mention. Okay. So how we are deciding?
Next session is time.
Sure. Okay, another 3 slides. Okay. So, your powder should be another 5 minutes only. Okay, please. So, your working capacity will be big and small. Okay. So, suppose you are adding the 500 gram of the powder, 500 gram, not 500 kg, 500 grams. Okay. So, you are going to add for the 500 kg. Okay. So, different will be there. So, how we are have to different bulk density of the product. That powder will be, how much density will be there, how to calculate. Okay. Suppose you have to take the powder for 50 gram, you are pouring to the measuring cylinder. Okay. So we are pouring to the measuring cylinder, some volume will come, correct? So, for example, 50 gram we are taking, how much ml is there? I mean volume, correct? For example, 50 gram divided by 96 or 56 ml. So, some value will come, 0.4 or 0.5, 0.6. Okay. That quantity, that value, you have to calculate with a different capacity. Okay. I am taking the 50 grams of the material 2 liter. I am going to take 5 kg or 20 kg or 100 kg. How we are deciding mean based on the bulk density of the powder. Okay. So, bulk density how to calculate? How much gram we are taking the powder divided by the how much ml we are getting. Okay. So, we have to calculate like that way for the RMG machine as well as coating machine also. This is for the RMG machine, rapid mixer granulated. So, this is a blender. Okay. Blender, how much capacity will be there? Like for example, 2 liter, 5 liter, we are going to the plant 400 liter, 600 liter, minimum is a 400 liter, maximum is a 1200 liters. Okay. That much powder we can mix. So, same like that way, how to calculate the volume of the material. Same like that bulk density. Okay. So, that is the reason only I told you calculation is a very, very important. Okay. So, we have to calculate based on your material quantity. So, this is the coating machine. So, coating machine, R&D machine this side and plant machine that side. Coating machine we can go one person inside and come back. That much big machine for the plant. Okay. If your R&D means only 2 liter or 1 to 3 kg we can use with manually. Okay. Machine will be running, we can take manually sample, we can see the physically as well as for the weight checking purpose. If you are going the plant, so sample purpose we have to take those, that much distance will be there, you have to take with rod. Okay. So, that much big capacity of the machines. So, we have to calculate here is that bream volume. Okay. So, bream volume how much will be there. So, we have to how much tablet you have to load, how much coating material you have to spray. So, everything have to calculate. Okay. So, these things and all easily we cannot learn. So, practically you have to learn minimum 2 months, 3 months it will take. How much quantity you have to take, how much you have to loading the material. If you are loading material, how much loss will come. Everything should be have to clearly have to mention in the BMR. BMR means batch manufacturing record. Okay. So, these are the book have to refer. Okay. So, these are the very fundamental things book. First one is a Lachman Lieberman is a Indian for international author, but it should be read properly. Okay, you can understand. But Remington is a international author, but if you are read means it is stable. I mean, it is a lifelong. It will be not forget. Okay. So, but is a reading is a very hard to read for the Remington. Lachman Lieberman we can read. Okay. Very easily you have to read. Okay. So, a simple example, these are the books you have to read for whenever the going the interview. Brahmangar book also will be there. So, whenever the going interview, please read this book. Okay. Minimum things you have to read, then you can go the interview. Okay. Okay. Thank you.
Shortage of time.
Okay, sir. No problem.
Thank you for your patient listening, and a big thanks to sir for his enlightening this session with a lot of information on generic product development. If any questions are there, sir will be available here for a shorter while. You can personally interact with sir. And due to time constraints, let us proceed to the next session. Thank you. Thank you to the management, to the principal, and to Mr. Rajaguru sir for sharing this wonderful information. Thank you all. Thank you, dear participants.
Thank you, sir, for your enlightening talk. As a token of our heartfelt appreciation and to commemorate the presence of our esteemed resource person, we now take this opportunity to present mementos to our speaker. I request Mr. R. Rajaguru, Manager, FRDQ Rafia Pharmaceutical Limited, R&D Center, Dar es Salaam, Tanzania, East Africa, to kindly come forward to receive the memento. I also invite the chairperson, Dr. A.S. Manjula Devi ma'am, to present a memento to Mr. R. Rajakumar sir to do the honor and present the memento. Now I invite Dr. R. Sambat Kumar sir to honor our chairperson with a shawl and memento. Thank you, sir. Let's have a tea break. Kindly come back within 15 minutes. Thank you.
போது நான் டீ சாப்பிட்டு வந்து மிச்சத்தை பார்த்துக்கலாம்.
இது முச்சுதான் மறு வேலை.
பொதுவா இப்படிலாம் வேலை பார்க்க மாட்டியா? இன்னைக்கு நான் தீயா இருக்கிற
தலைவருக்கு பண்றோம், தீயா இருக்கணும்ல.
அதெல்லாம் கரெக்டு தானா? ஆனா வைபே இல்லாம எப்படின்னா?
வைப் தானே.
ரைட்டு.
எதுக்கு பேச்சு ஒழுங்கா உட்காரு, எனக்கு பாச கிடையாது. எனிமி தோசை எவனா இருந்தாலும் முறைகளாமா சத்த முடிச்சிடலாமா. மல்லி மயிலாட்டம் பம்பரம் கட்ட சிடுபட்டா சே பல்லாட நம்ம ராஜா வெள்ளாட ரிங் தான் நம்மோட ஹே ஹே ட்ரம்பட் என்ன சே 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 Walking slow-mo மனசுல சோகம் Hey my golden sparrow
Bama bama kaman singu Queen teddy bandang kingu Daila daila pipi dumme Mala mati podu ringu Golden sparrow ya nengila arrow Neela the life ya romba sorrow Mask of sorrow nama payan da hero Singing duet vera yaro Kapila kapila gole iva singala sutana gale Singadi enna kele ini vanda aale nitili nitili meena oru gedula sutana neene nikadu nikadu line nee menakara moone golden sparrow ya nenjila arrow neela the life ya romba sorrow mask of sorrow nama payan da hero singing duet vera yaro இருக்கவா தினம் தினம் துடிக்கவா குவை டா இருக்குன்னா பிடிக்கவா இலக்கணம் படிக்கவா
Starry night till now, one story title na மானம் பார்க்காத மனாகுற ஏய் நானே போயட்டு எங்க என் டூயட்டு ரொம்ப நாளாக நான் தேடுறேன் ஏடி என்ன எங்கடி அடிச்ச ஏடி எட்டி கிடக்குற ஏடி என்ன எங்கடி தொலைச்ச ஏடி நானும் முழிக்கிறேன்
சொல்லிடு சொல்லிடு ஓகே நான் வைக்கிறேன் வெத்தல பாக்க கண்ணல கண்ணல லாக்கே இது கல்யாண சோக்கே
மின்னலு மின்னலு ஃபிளாஷ் உன்னை கண்டத நெஞ்சுமே கேஷ் உள்ளதும் போச்சே கதை திண்டாட்டமாச்சே
கோல்டன் ஸ்பாரோ என் நெஞ்சில ஆரோ நீ இல்லாத லைஃபே ரொம்ப சாரோ மாஸ்க் ஆஃப் சாரோ நம்ம பையன் தான் ஹீரோ சிங்கிங் டூயட் வேற யாரோ
Bama bama குயினை தேடி வந்தான் கிங்கு டைலா டைலா பிபிடுமே மாலை மாத்தி போடுறீங்க
கோல்டன் ஸ்பாரோ என் நெஞ்சில ஆரோ நீ இல்லாத லைஃபே ரொம்ப சாரோ மாஸ்க் ஆஃப் சாரோ நம்ம பையன் தான் ஹீரோ சிங்கிங் டூயட் வேற யாரோ
என்ன ஜாதின்னு சொல்லுங்க இல்லாட்டி குடுக்க சொல்ல மிக்ஸ் ஆகிடும்
கஞ்சி பட்ட தண்ணி ரமரவா குச்சாம் பண்ணி
குண்டா அடகு வச்சு சாராயத்தை புள்ளா குச்சி புட்டாட்டிய முஞ்ச வச்சு கத்து கதலை பமச்சி
துட்டு இருந்தா கஜா பிடி துட்டு இல்லாட்டி துண்டு பிடி
கிஞ்சா அச்சிக்கின்னு வலி வலின்னு வச்சிக்கின்னு டக்கி பெற வாங்கிக்கின்னு உடம்ப வீணா கெத்துக்கின்னு
கண்ணாலந்தா பண்ணிக்கின்னு சட்டா போயும் வச்சுக்கின்னு
தட்டிய கைவி விட்டு புள்ளிங்கள மறந்து புட்டு இருந்தாடா பக்கம் கெட்டு போயிட்டு மனம் கெட்டு
பல்லான கையிலேக்கா அது லுக்கு முடு சோக்கா உன்னை ஆக்கிடுடா பேக்கா நீ கைட்டி கடானேக்கா ஒன்றுங்கள கேவலமா என்னாதே மச்சு உன் கூட வந்து பொறுதலையா காத்து நேச்சு நல்ல பொண்ணு தேடி வரும் முன்ன
Those who are sitting backside, please come to the front. Those who are sitting backside, please come to the front. Front seat occupy பண்ணுங்கம்மா வாங்க. எந்த காலேஜ் ஸ்டூடண்ட்ஸ்மா? எந்த காலேஜ் ஸ்டூடண்ட்ஸ் பின்னாடி உட்கார்ந்து இருக்கிறது?
வந்தாலே சர்டிஃபிகேட் கொடுத்துருக்கோம். அந்த பொண்ணு வந்து எப்படி ஸ்டாஃப் ஸ்ட்ரீமில் சைன் பண்ண முடியும்? அதுதான் காலையிலேருந்து பிரச்சனை. ஐடி மாறி மாறி